Hi everyone, thanks for coming. Today I'm gonna talk to you about our work Global Transcriptome Analysis Reveals Circadian Control of Splicing Events in Arabidopsis thaliana. My name is Andres Romanowski and I did this work together with Ruben Schlein, Soledad Perez Santangelo, Estefania Mancini, and this work was performed in Marcelo Janowski's lab at the Fundación Instituto del Eluard in Buenos Aires, Argentina. So first, a brief introduction. What are circadian rhythms? As you know, we live in a world that cycles around its axis with a period close to 24 hours, and this leads to changes in environmental variables. For example, the most evident one is the day and night cycle, but we also experience changes in humidity, UV radiation, and temperature, for example. All of these changes have led to effects on the evolution of all the organisms that have developed on this earth. And this led to the development of an internal timekeeping mechanism that allows organisms to predict these environmental changes. This system is the circadian clock. So why circadian rhythms in plants? Well, chronobiology was actually born in plants. In 1729, a French astronomer named Jean-Jacques Tortus de Marran looked at Mimosa pudica plants which have leaves that open and close with the day and night cycles. However, he found out that if you put this plant inside a cabinet, outside of the reach of any external environmental cues, the plants continued to open and close its leaves with a period close to 24 hours. This led him to believe that there was an endogenous mechanism that was capable of sustaining the leaf movements. Since then, most of the circadian clock components have been elucidated, and a very basic schematic can be seen here on the right. That's the circadian clock of Arabidopsis thaliana. And then in the center, we can see different behaviors, if you allow me to say that word, in plants, or different outputs that are controlled by the circadian clock, such as, for example, gene expression, flowering, protein phosphorylation, stomatal opening, hypocotyl length, and cotyledon and leaf movement being one of the more classic ones. So this work was performed, as I said, in Marcelo Janowski's lab, and here I worked on many different projects, but the one that I'm telling you about today is the one that is highlighted here in green, which is the global circadian control of alternative splicing. So we already knew when we started this project that the clock controlled many co- and post-transcriptional processes, such as, for example, transcription, right? We also knew they controlled poly-A tail length, mRNA degradation, and nuclear export, and we also had some evidence of alternative splicing control of, for example, clock components, right? So what's alternative splicing? So this is the process through which a gene can lead to many products, by the alternative splicing of different introns and exons that are contained within the genes. So in constitutive splicing, these introns, which are the blocks that are in between each of the exons, are completely removed. However, other stuff can also happen. For example, an exon can be skipped, like in the second example, where exon 2 is removed, right? An intron can be retained, right, like in the third example. Or, for example, other splicing sites can be used. So if an alternative 5' splice site is used, you can see that in example 4, and in the last example we see when an alternative 3' splice site is used. There are other combinations, but they are beyond the scope of this brief introduction. So our question is, what's the extent of the circadian regulation of alternative splicing? To answer that question, we performed the first Arabidopsis thaliana global circadian mRNA-seq. We grew plants in 12 hours photoperiods, and after 12 days we transferred them to continuous light. On day 2 of continuous light, we sampled every 4 hours for a total of 48 hours. We extracted the total RNA, we did our RNA-seq, and the first thing that we looked at was, of course, gene expression. And as we can see here, 9,127 genes were found to be rhythmic. On the lower left, you can see a heat map where yellow always indicates high expression and blue indicates low expression. And we can see how these genes go up and down along the 2 days of these circadian cycles. Also, In order to validate our approach, we perform qPCR of the usual suspects, and you can see here 4 different clock genes whose pattern very closely match that of the RNA-seq traces, which are drawn in gray. So using this approach, we found several novel genes when we compared our dataset to those that were published previously. So one thing to take in account is that some of these datasets, well, actually all of these datasets, were performed using microarray technology, right? So many genes might not be available in using those technologies, whereas in mRNA-seq we can capture all of the transcriptome of Arabidopsis. So when we compared our dataset to those previous datasets, we found 3,545 novel cycling genes, and most of these genes had to do with RNA processing and translation, as you can see in the GO-term analysis on the right. We also saw that in our novel cycling genes, there were 146 transcriptome factors that were not known to cycle before. You can see that on their pattern of expression in the heat map on the lower right. So this will lead to novel avenues of research. However, we were not focusing on gene expression. Our original question is, does the clock globally control alternative splicing? Well, that's not so easy to answer, as usually splicing analysis revolves around using isoform reconstruction. This isoform reconstruction of those very short reads of due to mRNA sequencing can lead to several biases. So we wanted to overcome those biases, and to do that we used a bioconductor package that was developed by Aspli, who was at the time a PhD student of Marcelo's while I was a postdoc there. So using her particular package, what we can do is, instead of doing an isoform level analysis, we do a splicing event level analysis. So we analyze each of the events. So the software bins the genome, as you can see on the left, on different exonic and intronic bins, and what we do is we quantify the reads that match those particular bins. Okay? So we normalize these bins to the gene expression so that we don't have an effect due to gene expression. We perform ANOVA-like and read density filters. We filter by an FDR value below 0.1 and a p-value below 0.05, and then we check for rhythmicity using JTK cycle. So to cut the story short, using this particular pipeline, we found out that yes, the clock does control alternative splicing. You can see the heat maps here, and I can tell you that we found 1,513 clock-controlled events, or CCEs, that corresponded to 1,169 genes. You can see the pattern on the right, along the 2 circadian cycles that we analyzed, and then we quantify them using semi-quantitative RT-PCR and SDS page analysis. You can see those on the right, and those match the pattern on the left. Here you can see read density histogram of this particular exonic bean that we're looking at. Actually, it's an intronic bean, and you can see all the different samples on the y-axis. And what we can see here is that at the beginning there are no reads that map to this intron, and as we go along the cycle we see that the intron starts being retained, then it goes down again, and then it goes back up again. And on the right you can see the intron retention ratio, which is the analysis of the SDS page. Okay, so now that we determined that the clock does control alternative splicing, we reasoned, okay, so there must be some splicing factors behind this. So we intersecting our list of the robust cycling genes, which are those from our RNA-seq, but that also cycled in those previous global analysis that I mentioned, and we intersected that list with a list of splicing-related genes. One of these 31 genes that were clock-controlled was an homolog of survival motor neuron. However, when we did a phylogenetic analysis and closer inspection of the sequence, we found that it more closely matches SPF 30, splicing factor 30, which is required for spliceosome assembly. So we obtained two different tDNA mutants of SPF 30, we called 1 and 2, and we assayed different clock-controlled outputs, classic clock-controlled outputs, to see if there were any effects due to the lack of this splicing factor. So when we looked at leaf angle, we found no differences whatsoever with wild type, but however, when we looked at flowering, we saw that splicing factor 30 mutants constitutively flower earlier than the wild type counterpart. But what happens in terms of splicing? Well, we did an mRNA-seq of splicing factor 30 mutants, and we saw that it mostly affects intron retention, as you can see in the lower left in panels A and B. When we looked at the splicing events that were both affected by SPF 30 and controlled by the clock, we saw an overlap of 32 events. Yeah. As splicing factor 30 mainly affects intron retention, we reasoned that it could be affecting overall retention levels along the whole circadian cycle. And in effect, if you look at panel E, one example event that's both clock controlled and controlled by splicing factor 30, yeah, we see that if we compare that wild type line, the black line, to the blue line, yeah, the one that corresponds to splicing factor 30, there's in effect a higher retention of the intron along the whole circadian cycle. So in summary, environmental cues control and train the circadian clock, and the circadian clock controls gene expression and also alternative splicing. There are 617 genes that are both controlled at the gene expression level and also the alternative splicing levels. One splicing factor that is also clock controlled is splicing factor 30, which mainly affects intron retention, and it also controls a subset of those clock controlled events. This work has been published in the Plant Journal and has been awarded with the Best Resource Article of 2020. If you have any questions, please feel free to drop me an email or contact me via Twitter. Thank you very much for your attention.